Hey, all right, you want to go ahead and get out all of the cellular respiration notes, and I'm actually going to redraw this for you, and you pay attention and learn from this. Take extra notes or maybe draw it again. Just watch and make sure you know all of this. Okay, I'm going to try to draw it with my finger. Okay, so this is cellular respiration. And it happens, here is my mitochondria. Okay, so the first thing that we got is glucose is split. And remember, glucose is 6 carbons. So 1, 2, 3, 4, 5, 6 carbons of glucose are split. So this is glucose. And it's split in two, and it makes 2 G3Ps. But then eventually it becomes pyruvic acid. And remember, this process takes negative 2 ATP. So it required 2 ATP. But you actually made 4. So we say there's a net of 2 ATP and 1 NADH from step 1. And remember, step 1 is called glycolysis. And I don't have room to write it, but step 1 is glycolysis. Couple facts about glycolysis. Glycolysis happens in the cytoplasm. Glycolysis does not require oxygen. So no oxygen happens in the cytoplasm, and all organisms do this. So this leads to common ancestry, that all organisms do it. Okay, so the pyruvic acid then is going to go into the Krebs cycle. So step 2 is the Krebs cycle. And the pyruvic acid goes in. And pyruvic acid, which was 3 carbons, right? It was 3. Let me go ahead and draw those. It was 3. 1, 2, 3. But it was, so 1, 2, 3 is now going to be split. Oh no. Is now going to be split and it'd be a 2 carbon molecule. So the pyruvic acid becomes a 2 carbon molecule called acetyl coenzyme A. So pyruvic acid becomes acetyl coenzyme A. And that actually happens a little bit before the Krebs cycle. And then that acetyl, so that's like at the very beginning or right before the Krebs cycle. And then the acetyl coenzyme A goes through a series of events. And those, at the end of the Krebs cycle, so, and remember in the notes, when I originally drew it, I just kind of made a cycle like this. So, and then at the end of the Krebs cycle, you've actually made, so remember the Krebs cycle has to happen twice. So Krebs cycle times 2. At the end of 2 turns, because you've got 2 pyruvic acids, at the end of 2 turns you have 6 NADH you have 2 FADH. And remember, these are electron carriers. I actually remember in class I called them school buses. So you have 6 NADH and 2 FADH and 2 ATP. Okay, also this carbon right here was released as CO2. And that's when you exhale. You release CO2 when you exhale. Okay, let's talk about the difference in NADH and FADH before we go any further. NADH and FADH are both electron carriers, and they both carry 2 electrons. The difference is NADH makes 3 ATP, and FADH only makes 2 ATP. And the reason you're going to see in a second, but the reason is because NADH drops its electrons off at the beginning of the electron transport chain. FADH drops its electrons off at the second electron molecule in the electron transport chain. So an easy way to say this is that NADH actually makes 3 hydrogens go into the hydrogen concentration. And that makes 3 ATP. FADH only makes 2 hydrogens go into the hydrogen concentration. So that only makes 2 ATP. So let me draw in and I think you'll understand better. Okay, so remember the Krebs cycle happened inside the cristae, on the inside of the cristae. So my cristae, I'm just going to draw right here, and that's folded to increase surface area, but just for room's sake, I'm just going to put it on this side, and it's only kind of folded. Well, I want to draw it a little smaller than that. Okay, so NADH comes and drops its electrons off. Let's go with green for this. Drops its electrons off right here at the beginning. So it drops its electrons off. NADH drops off electrons or loses electrons. So we could say that NADH is oxidized because oxidation is loss. Okay, so NADH we just said is oxidized because remember oxidation is loss, reduction is gain. Okay, so NADH drops its electrons off at the electron protein right here in the electron transport chain. So here's my protein. I'm going to draw this out. Here's another protein. Here's another protein. And then here is ATP synthase. So ATP synthase, I'll outline that one in black, so it's kind of important. There we go. There's ATP synthase. Okay, so the electrons go down the electron transport chain, and remember, this is a phospholipid bilayer. So there, I'll draw the little phospholipids. There we go. Those dots are phospholipids. Just pretend it's small here. Okay, so the electrons in green are going down the electron transport chain, and they bind with what? You know, they bind with oxygen. And that's going to make, inside the mitochondria, that's going to make water. When 2 hydrogen electrons bind with oxygen, that makes water. So we would say that oxygen is reduced. NADH was oxidized. Oxygen is reduced. So the electrons go down the electron transport chain. Well, as they go down the electron transport chain, you know what's happening. Hydrogen molecules are pumped in. Oh no. Hydrogen molecules are pumped out of the cristae into that space in between the cristae and the mitochondria. So the hydrogen molecules, let's see if we can make those arrows a little smaller. Hydrogen molecules are pumped out here into that confined space, and it builds up a hydrogen concentration gradient. So you got a whole bunch of hydrogens out there. Then the hydrogens are going to go through ATP synthase. And when they go through ATP synthase, that's going to make ATP. So ADH binds with the phosphate and makes ATP. So ADP, I think I said that wrong. ADP binds with the P and makes ATP. So total, at the end of it, you've got 32, maybe 34, but 32 or 34 ATP, 2 and 2, all from one molecule of that first glucose. So that's the 3 steps to cellular respiration. Okay, let's talk about location. The location of glycolysis, which was step 1 in the cytoplasm. The location of the Krebs cycle is inside the cristae. The location of the electron transport chain is on the membrane of the cristae. Okay, let's talk about oxygen requirements. Glycolysis does not require oxygen. The Krebs cycle does require oxygen, and the electron transport chain does require oxygen. So the glycolysis is the only one that doesn't. Okay, step 1 and step 2 make ATP by breaking a substrate. They make ATP by breaking a substrate. So 1 and 2, there's 2 and there's 1, we call those oxidative phosphorylation. So oxidative phosphorylation. I don't have enough room, but you get it. Okay, the third one is not called oxidative. Oh no, I totally messed up just now. I'm so sorry. The first and second steps, wow, that was bad. Bad, Miss Gilbert. The first and second steps are called substrate level. Substrate level phosphorylation. Man, that was bad. Okay, let me explain why. Because step 1 and step 2 are both breaking a substrate to make ATP. So you're breaking a substrate, phosphorylate, to make ATP. So step 1 and step 2 are substrate level phosphorylation. Okay, so let's take that off my pretty picture. Okay, the third step is making ATP because NADH was oxidized. NADH was oxidized. So the third step we would say is oxidative phosphorylation. Oxidative phosphorylation. Okay, so steps 1 and step 2 are substrate level phosphorylation. Step 3 is oxidative phosphorylation. Okay, let me add one more detail to my picture, and then we'll move on to cellular respiration part 3. Okay, NADH, we said, drops its electron off right there at the beginning. Well, FADH, if you'll look at this dark blue arrow, FADH drops its electron off at that one, at that molecule. Right there. And so when the electrons go down, FADH only produces 2 hydrogens, so it only makes 2 ATP. So look down here at this side. NADH makes 3 ATP, and FADH makes 2 ATP. Alright, so hopefully this was helpful. Go ahead and get out cellular respiration part 3 notes, and we'll start there. Okay, so you probably already figured out just by looking at the notes. And remember, cellular respiration part 3, just kind of by looking at it, this is going to be anaerobic respiration, or respiration without oxygen. Okay, so things you should already know. I'm actually going to make a blank page here. Okay, so things you should already know. All cells, prokaryotes and eukaryotes, do glycolysis. So we already said that in the last video, or in this video, but earlier. Since all organisms can perform glycolysis, they must be united by common ancestor. So because all organisms do glycolysis, that leads people to think common ancestor. Okay, the third thing, enzymes control most processes within cells. Therefore, they must be regulated or controlled. by inhibitors. And y'all remember the difference in, remember we drew this out, the difference in a competitive inhibitor blocks the active site. And a non-competitive inhibitor blocks a site other than the active site, like the allosteric site. But both are going to slow down the rate of production. Okay, so if no oxygen is present within a cell, you got one of two choices. Once glycolysis happens, it's either going to do alcohol fermentation or lactic acid fermentation. Okay, so glycolysis happens regardless of whether there's oxygen or not. So after glycolysis, if there's no oxygen, then the cell has to do either alcohol fermentation or lactic acid fermentation. So number 1 on your note says if no oxygen is present, that's going to be anaerobic. And then it defines fermentation. It says fermentation will occur to free up electron carriers. Electron carriers are NADH. So free up electron carriers to keep at least glycolysis going. Okay, so the first one is going to be alcohol fermentation. And so here's what's happened. After, I'm going to actually draw glycolysis one more time. So 1, 2, 3, 4, 5, 6. Glycolysis splits that glucose molecule right down the middle into pyruvic acid. Okay, and remember, this makes 2 ATP, net of 2, and 2, 1 NADH. Okay, so now there's no oxygen, so the Krebs cycle can't happen. So no Krebs cycle. Either alcohol fermentation or lactic acid fermentation are going to happen. And luckily for us to remember, they're really similar, so it's not very hard to remember. Okay, so this pyruvic acid is going to be made into either alcohol or it's going to be made into lactic acid. Okay, and in the process of that, all it's going to do is make NADH, and I'll change colors here. It's going to make NADH become NAD+. Remember at the beginning right there, it says fermentation will occur to free up electron carriers. So what it's doing is it's just taking the electrons from NAD+, so that when this is freed up, glycolysis can happen again. So you're getting 2 ATP, glycolysis is happening, So it says fermentation. They convert the 2 pyruvates into 2 molecules of ethyl alcohol, or ethanol, same thing, by cutting off CO2 and filling the open bond with hydrogens from the electron carrier. So they take the hydrogen, so you do both pyruvates. They fill up, they take the CO2, it says, take the CO2 and remove the electrons from NADH. And then, this is important to highlight, this frees up the electron carrier to keep glycolysis going. So it frees up the NADH to keep glycolysis going and make some ATP. So whether you make ATP with alcohol fermentation or with lactic acid fermentation, that depends on what kind of organism you are. Okay, so for example, yeast is going to do alcohol fermentation. Okay, Some bacteria do alcohol fermentation. So this is just an example. You take the bacteria and you take strawberries and a lot of sugar, and don't try this, but if you cut the oxygen off, then the bacteria is going to eat the strawberries. Without oxygen, they're going to make alcohol as a byproduct. So they're going to release alcohol as a form of their natural waste. Okay, and that's how to make strawberry wine or some sort of other wine. If you do that same process with potatoes, so you get potatoes and you cap the oxygen off, take the amount of oxygen out, put it with bacteria, and it may be yeast, I'm not sure. So it just depends on what organism. Some organisms do alcohol, some organisms do lactic acid. You take the potatoes and you take the bacteria, cap it off, and that's actually how vodka is made. So the bacteria eat the potatoes, and that releases alcohol as a form of lactic acid. And again, don't try that because there's lots of things that you have to do just right in order for it to be okay. So you don't want to try that. Okay, lactic acid fermentation, so this is going to be the second one, is mainly in animals. You make lactic acid when you don't have enough oxygen, not in your brain, but in your biceps, per se. So if you're doing bicep curls and you're working out and you keep on curling your arms, working out, well, oxygen is not going to be in your bicep anymore. You're still going to be breathing oxygen, but the oxygen may not be stored in the muscle cells because you've used it. So when you're working out, you're using oxygen in the cellular respiration process. You're using oxygen and you're making that ATP. Well, when there's no more stored oxygen, in order to continue getting ATP, your body takes the pyruvates, and it can't do the Krebs cycle. So it takes the pyruvates and it makes lactic acid, converts pyruvate into lactic acid by breaking the ketone, the double bonded oxygen in the middle, and adding hydrogen. The hydrogen comes from that NAD+. And you make lactic acid. So there's lactic acid. Okay. It says here again, keeping glycolysis going. So glycolysis. That's why you get out of breath when you're working out, because your body's trying to get more oxygen to your muscles. Okay, it says cheese, yogurt, and muscle cramps. So this is how you make cheese. This is how you make yogurt, and this can give you cramps. They're all created by lactic acid fermentation. Okay, so the next thing, if you're looking at the notes, the next thing says facultative anaerobes. And those are organisms that can perform both. They like to have oxygen. They prefer to have oxygen. But without the presence of oxygen, they can survive. And that would be the way to be if you could. Like, oxygen would be nice, but don't have to have it. Can survive without it. Okay, we actually already talked about glycolysis and the significance to evolution. It says early Earth had no oxygen. So 3.6 billion years ago, there was no oxygen. So glycolysis was the only way for those bacteria to make energy. Like all organisms do glycolysis, which points to common ancestry. And then take a second and review endosymbiosis. That's where one bacteria was engulfed into another bacteria. And that small bacteria may have evolved into a mitochondria or a chloroplast. Okay, amino acid utilization. Okay, so take a look at this picture. The cellular respiration that we've been studying is with sugar. So we actually have been studying how the carbohydrate makes sugar, so there's your carbohydrate. It makes the pyruvic acid, goes glycolysis, acetyl-CoA. Oh, citric acid and Krebs cycle are the same thing. You may see citric acid, so know that. And then oxidative phosphorylation is, of course, ETC. Okay, but sometimes if you don't have carbs, you would break down stored carbs, of course, but your body can get energy from, so look right here, it can actually get energy from fats or from proteins. Okay, so let's look at the notes. The first thing is amino acid. So remember that this is an amino acid right here. You got the amine group on the left, the carboxyl group on the right. You should be able to draw that by now with your eyes closed. And then the R group in this picture is on the top, but when I draw it, sometimes on the bottom. Okay, so look, what if we need to break down a protein? So read with me in the notes. It says amino acid utilization. That's going to be breaking down proteins to get ATP. They must undergo deamination. That means the removal of the amine group. Look, the removal of the amine group. That's deamination. Cut off the amine group and put coenzyme A right there. So cut off this amine group and put coenzyme A. And that's how it enters the Krebs cycle. It says using a sulfur molecule to the remaining 2 carbon skeleton from the amino acid. Then feed into the Krebs cycle to make energy. So the resulting is NH3, which is ammonia. Right? Y'all see NH3? That's ammonia. It's put into the blood, and that's urine. So NH3 is mainly what you urinate, or uric acid for birds or reptiles. Fish release the NH3 directly into the water. So yours is a little more diluted. Yours is urea. Okay, so when you see that this is on the test, so make sure you're paying attention. Make sure you're paying attention. When you break down proteins, instead of sugar, so we just drew it out with sugar, but when you break down proteins, it's called amino acid utilization. Okay, and that's where you take the protein and look, it goes right here, and there's your NH3, breaking off that amine group. It goes into the Krebs cycle. Remember, that's the Krebs cycle. And then electron transport chain. Okay, if it's a fat, it's called lipid utilization. Okay, make sure you're listening. This is big. These undergo, it says beta oxidation. Oxidation means the loss of electrons. Okay, so it says beta is the second letter of the Greek alphabet. Okay, so looking at this fat right here, cut the fatty acid tails off of the lipid molecule. Cut the fatty acid tails off of the lipid molecule. And so now it's 2 carbon skeletons, and attach coenzyme A here. So attach the coenzyme A here. Using a sulfur molecule to each 2 carbon skeleton. Then feed that into the Krebs cycle. So if it's beta oxidation, that's the lipid. Oh, here we go. Oops, there it is. Lipid right there in the fats. Okay, biosynthesis. This is, okay, so we break down sugar and we make ATP. But we do that so that we can make, let's go back to this picture. We all have to do this. We have to make ATP so that we can build up. You have to build up 2 macromolecules with anabolism. You have to build them up, right? You have to build protein in your body, and you have to build DNA in your body. So it says this is using food building blocks to make yourself, basically anabolism, which requires ATP. Okay, last picture here. feedback inhibition, we've actually talked about this. Feedback inhibition. That's where the product, so write this down, the product of a reaction, so feedback inhibition, the product of a reaction stops, let's say, the product of reaction acts as an inhibitor and stops the reaction from occurring. Okay, look at this picture for a second. You can see you got glucose breaks into the pyruvate, goes to the ETC. So this is cellular respiration in a pretty simple form. Glucose goes to the electron transport chain. Okay, well, when you make plenty of ATP, and you're just still sitting on the couch being lazy like you are, ATP, look right there, you see it? ATP is going to go up and it's going to stop glycolysis from occurring. So read what you wrote again and make sure you understand it. The product, aka in this situation, ATP, the product of the reaction acts as an inhibitor, and it's going to bind to that initial enzyme. Remember, inhibitors, think about it. It acts as an inhibitor and stops the reaction from occurring. So your notes say the enzyme phosphofructokinase, you know that word, acts as the on-off switch for glycolysis. When there's plenty of ATP, excess ATP from the Krebs cycle will work as co-inhibitors attaching to an allosteric site to shut down the whole process. So if it attaches to the allosteric site, is it a competitive or is it a non-competitive inhibitor? And if you're thinking non-competitive, you are correct. Okay, go ahead and put your notes away. The sub is going to pass out today's math quiz. Now, listen to me. Before you don't move, listen. This math quiz is hard. There's something, you've never done this before. You're going to need your formula sheet. You're going to need your calculator. And if you think you can answer this question and get it right, but you're going to have to use your brain and use the formulas on the math quiz. I went ahead and put it on there in case you couldn't find your formula sheet. But if you want to use your formula sheet, you can do that too. But all this is simple conversion. You did conversions like set up the railroads. You did conversions in chemistry. It's not that hard. You're just going to have to think. Okay? If you can't answer it, it's okay. Just turn it into the basket. We'll work on it on Monday. Don't talk, though. This is an individual math quiz. Now, the synthesis question's on the bottom. Answer that, just like always. And when you finish with your math quiz, you turn it into the basket, and you're going to get the opportunity to work on your prezzies in class. So you still have several minutes. You can work on your prezzies in class, or finish your lab questions from yesterday. Not the lab, but you can finish your lab questions from yesterday. And I will see you Monday. Email me if you have any questions.